Here we go. It's part 2 now of the central nervous system. We're looking at the brain, and we just got through doing the meninges and the cerebrospinal fluid flowing through the system here of ventricles and subarachnoid space. And I'm just going to do a quick review to make sure that we have covered all the details in absolute detail, shall we say, with great integrity. So what we have here is the left and the right. There we go. The left and the right lateral ventricle. 1, 2. Here's the third ventricle. The way from the lateral to the third ventricle is through the foramen Monroe. From the third to the fourth, we go through the cerebral aqueduct. Note that the fourth is the end of the trip unless we're going to the central canal of the spinal cord, right? Otherwise, there are apertures for exit. There are 2 lateral apertures, and L is also the same first letter of Luschka. And so we have the foramina of Luschka left and right. And then we have the letter M for median aperture, which is also the first letter of Magendie, the foramen of Magendie. Count them 1, 2, 3. The 2 foramina of Luschka on either side, left and right, and the central median aperture, which is the foramen of Magendie. Where do these go is the next question. Let's take a look here. And where it goes here from the fourth ventricle is to the subarachnoid space. Okay, where does the cerebrospinal fluid come from? It comes from the choroid plexus of capillaries in the lateral ventricles, in the third ventricle, and in the fourth ventricle. What is the procedure in flow is from lateral to third to fourth to subarachnoid. There's also proceeding down through the central canal of the spinal cord. Once we are subarachnoid, which means we're between the pia mater and the arachnoid membrane, we are subarachnoid. We are surrounding both the brain and the spinal cord. And the brain and the spinal cord are literally floating in cerebrospinal fluid, even as we sit, walk, stand, sleep, etc. Our central nervous system is floating. How does the cerebrospinal fluid get back? It gets back to the bloodstream, or from whence it has come, through the arachnoid villi, diffusing into this unit right here, the superior sagittal sinus. Okay, so the dura mater is 2-layered in the brain, and they separate very neatly at certain places so that we can have an opening for venous blood flow. The venous blood flow in the brain, throughout the brain, is always from front sweeping back. Okay, so we always have a backward flow, and we're going to be addressing that in Biology 20B in the blood vessels when we study in intimate detail the flow of blood from the heart to the arteries to the capillaries through the veins in back to the heart. So, and just to make sure that you understand meninges, we're going to have the pia mater right on the surface of the brain, a very thin, delicate layer, pia mater, the tender mother. We're going to have the next layer, which is the arachnoid membrane, the arachnoid mater. Okay, and so this is the spider mother right here, the tender mother, the spidery mother that has the dentoid ligaments that look a little bit like a spider web. And then beyond that, we have the 2 layers here of the dura mater. So the dura mater is two-layered. Okay, I will not test you on these words specifically, but you should probably know that there is a periosteal layer outside and a meningeal layer inside. Okay, they separate at times and form venous sinuses, and here's the superior sagittal sinus. And we have the arachnoid membrane is going to be actually sending little villi. Sometimes we call these granulations. Okay, but they're identifying it here as an arachnoid villus, and we get diffusion back into the bloodstream. Okay, so now I've fully reviewed that for you, and I hope that every detail of it is fixed in your mind. Okay, so we have a lot of territory to cover here. First the brain and then the spinal cord. And so as we proceed here to this next page, we're in part 2 of this central nervous system adventure. Here is an actual view of the brain. Now notice that over here, do you see that difference between this side and this side? This side is completely devoid of the vascular system as well as this shiny stuff here, which is the arachnoid villus. This is like saran wrap, right? It's a clear, see-through membrane, okay, that holds the cerebrospinal fluid in and also protects the cerebral veins and arteries, right? You remove that, sort of hose it down, clean it off, dry it out, and now you have this vision right here. Here is this central, how shall I say, division right here. This is a fissure that divides one side of the brain from the other. We have two completely unique brains in a sense in both sides. It's only connected by something that is referred to as a corpus callosum. We have 5 lobes on each side of the brain. 5 of them, count them. We have a frontal lobe, we have a parietal lobe. Frontal has things like intellectual reasoning and planning and so on. It has also motor activity for both eyes and the rest of the body. The parietal is a somatosensory region. It has memories of somatosensation. You remember what velvet feels like. Then we have a temporal lobe over here, which has to do with hearing and sound, and to some people there's language focused in there. Over here we have the occipital lobe, which has much to do with sight and the memories of vision. And then we have a 5th lobe where you have to actually go deep in here, and here they've done that for us. Isn't that nice? The artist has actually drawn in the retractors. And right here is the insula, a 5th lobe. So we have here the frontal, the parietal, the occipital, the temporal, and the insula. 5 lobes in each of these hemispheres of the brain. Okay? So, in fact, the brain is so elaborated, it's almost like trying to stuff a down sleeping bag into its stuff sack. We actually have to sort of crumple it all up and fold it up in order to fit it into the skull. That's how big our brain is. Okay, it's right up there. It's practically cancerous. So I want to also note here that when something bulges out, we're going to be calling it a gyrus. So here we have the term gyrus. Whenever we have an infolding, we're going to be calling that a sulcus. Along the edge here, we have the gray matter, which is where the cell bodies are and where the synapses are for the brain. And there is an elaboration of that. I want you to be very clear that we're born with only about 1/3 of that gray matter, and that in the next 5 years of life, outside the womb, ages 0 to 5 years of age, we are elaborating the other 2/3 of the brain. Put that in your mind. A lot of things are not learned. Do you understand? They are maturational. They come about as a result of grown circuitry. such as walking, is not learned, so don't torture your child to walk. Don't torture your child to talk. Talking is not learned. It is something that comes about as a result of increasing circuitry. We'll get back to that in our episode 3 here.